Hello everyone. This lecture is actually a combination of some key ideas in Chapter 6 and Chapter 7 that correlates how nutrition and growth and metabolism are all tied together. So of course enzymes are key in any metabolic reaction. They're the thing that actually helps us drive the reaction in the direction that we need. Now most metabolic reactions could go on without any help, but they wouldn't necessarily happen in a time frame that a living organism needs. And so enzymes are there to sort of play matchmakers and make sure that everything that can happen in this metabolic reaction does happen because they have the right three-dimensional fit or affinity. So what are enzymes? Well, primarily they're a protein, right? So the bulk of the enzyme is going to be a three-dimensional protein held together by hydrogen bonds that hold that long amino acid sequence in the proper folding pattern. Now some enzymes are going to have a coenzyme, which may be some other kind of organic cofactor, but typically when you put the cofactors, they're going to be another kind of substance, like an inorganic, like a heavy metal or a salt, like magnesium or something like that, is going to help complete this enzyme. And so if an enzyme requires cofactors, it would be referred to as a holoenzyme as long as those cofactors are present. So you can see how this spot up here at the top, this is the active site. This is where the substrate would fit. Sometimes they're going to call this the catalytic site because it catalyzes reactions. So that active site, you can see if you were missing either of those cofactors, would not be the proper shape. Now some enzymes also have another affinity site on the opposite side or just anywhere other than the active site where other cofactors could fit. This allosteric site is going to come into play here when we speak about inhibitors, things that can come in here and block the activity of these enzymes. So our current understanding of enzymes is that when the substrate comes along that fits, the enzyme actually changes shape briefly. They call it a conformational change. And so there's this model or this sort of a theory that describes how enzymes work, and it's called the induced fit, meaning it has the right shape, it will attract or have affinity for the right substrate. When those two find each other, the enzyme actually physically changes shape slightly to sort of latch on and have a tighter grip on that substrate to make sure the reaction happens, right? If these things are all just randomly colliding with each other, what's to keep it from just bouncing right off before it does its job? So the induced fit model means that enzymes are going to slightly change their shape when that substrate comes along, sort of latches on, completes the reaction, then it'll let go and keep going and repeating the process as long as there's substrates to work with. Recall there's other things that can help drive these reactions that we're talking about. You got to have the proper temperature. We talked about the preferred thermal profiles that each living thing requires. And so this one, clearly the preference here would be about 37 degrees or human body temperature. Now reaction rates are also driven by how many partners are present, right? Enzymes and substrates need to be found in equal concentrations or the reactions will stop. Now at some point you have, if all the enzymes are saturated, meaning they're bound to a substrate, then that enzyme activity will sort of level off. If substrate concentrations run out, you'll also sort of hit this leveling off or saturation point where the reactions will stop. One way that organisms can have real fine-tuned control over their metabolic reactions is by using compounds called inhibitors. Now inhibitors are going to be those things that block enzyme activity without actually damaging or denaturing those protein enzymes. So for example, if you recall what denaturing looks like, if you have a functional globular protein, one that is three-dimensional, that has a very particular shape that fits its substrate, this is what it would look like. Here's the little dot, dot, dot. If you recall, that's the symbol for hydrogen bonding. This is just showing you that you have affinity between the substrate and the enzyme. But if it's exposed to high heat or pH extremes, typically those hydrogen bonds can't tolerate that, and so the protein would denature and no longer have affinity for its substrate. So generally, if a cell has an inhibitor, it's going to fall in one of these three categories. Competitive inhibition is a very common way that organisms can control their metabolic reactions. So you can see here why they called it competitive. If you have a compound that has the same affinity that the substrate does for that active site, then if the competitive inhibitor is there in a higher concentration, it'll saturate those enzymes and keep the substrate that the cell would normally be using from operating. This is actually one of those sort of ideas behind pharmacology, right? If you're trying to drive a certain process, you would put that medication in the body and hope that it is binding to the right receptor in the right place, or shutting down enzymes you don't want to be activated, or activate enzymes you do want to be activated. It's all about concentration gradients. Competitive inhibitors work by outcompeting the substrate at the active site. Non-competitive inhibitors, on the other hand, don't bind directly to the active site. They work on that allosteric site. And so it's going to be found somewhere other than right there at the place where the substrate would bind. And so what happens when the allosteric inhibitor arrives, it's going to cause that enzyme to slightly change shape, and the substrate will no longer be able to fit in that active site. So either way, you've stopped the reaction. It's just doing it in a slightly different way. So most metabolism requires more than just one reaction. And so what you're seeing here is sort of the big picture. When you have a substrate that binds to an enzyme, the product of that reaction is probably going to be the substrate for the next reaction. And so what they're calling here the intermediate, right? It's the product of the first step, but it's an intermediate because it's immediately going to be used as the substrate for the next. So then another enzyme has affinity for that intermediate. It will convert the substrate into something else, and maybe another intermediate. And then eventually you get through all the steps required in this metabolic reaction, and you end up with the end product, which is what was the purpose of the series of steps in the first place. So once you have that end product at a certain concentration, it actually will come back and feed back to that enzyme at the very beginning of the pathway and do what we just saw with the non-competitive inhibition. So if you have enough of the end product present, you don't need to do that reaction anymore. And so it's a really effective way to basically shut this down. You wouldn't want to shut down enzyme number 2 or 3 because you might have wasted the energy required in the first couple of steps. And so it's really effective to go back to the very first step and shut that one down. Now, when that end product gets used up or it diffuses away, then it would come off that first one, and then the process would open back up and it would operate again. So they call this one feedback inhibition. It kind of includes that non-competitive inhibition that we just saw in the last category. So put it all together, basically metabolic reactions include both catabolic and anabolic reactions. Remember, catabolic is, I refer to the word cut because catabolic cuts. You're going to take nutrients in, break them down into their monomers. So if it's a protein, you break it down into amino acids, and then your cells would have to put them back together the way that they need. So maybe those amino acids would be put back together to make the proteins in the cytoskeleton or enzymes or something else. And so anabolism or anabolic reactions would then put those things together the way the cell needs. And once you've built enough macromolecules, then the cell can divide and continue on. Now realize no metabolic reactions are perfectly efficient, meaning most of the time you lose a little bit of energy in the process. You use ATP in the process to run those reactions, but you also lose some of the chemical energy as heat. Now in our bodies, we absorb that heat in the blood, in the water of the bloodstream, and so we can make our own heat and retain it. Microorganisms lose that to the environment, and so they have to rely on their culture conditions to maintain that temperature. So when an organism does catabolism, of course, glucose is the preferred energy source for most living things. There are some other microorganisms that use other things, but we're going to primarily focus on glucose. And of course, if you recall the recipe for glucose, C6H12O6, if we're going to break that down, we're going to do it in one of two ways. The most efficient way is what we call cellular respiration. This is going to completely break down that glucose molecule into carbon dioxide and water. So we're going to look at the major steps involved there. Fermentation is considered an incomplete catabolism because it's going to leave behind organic substances that generally are waste products to the organism, and they're usually in the form of either acids or alcohols. So recall we talked about bacteria. When they ferment, substrates will produce acids. When yeast ferment substrates, they will produce alcohols. So when we go through metabolism here, I want you to really focus on keeping this simple. Right, your book goes into a lot more detail on enzyme names and everything else. We're going to look at this process in the big picture. We're going to name the major steps, or what I call the pathways. We're going to identify the substrates that go into each of those. And of course, substrates go in, products come out. Byproducts are those things that also come out of a metabolic pathway but don't necessarily feed the very next pathway. So when we talk about a product, it's the thing that's moving forward in the process. Okay, so again, keep it real simple. What goes in, of course, is going to be, if we're doing cellular respiration, glucose is the most common thing. Glycolysis is the process we're going to do here. We're going to take a 6-carbon molecule glucose and split it down the middle. And we're going to end up with a 3-carbon molecule called pyruvic acid. Pyruvic acid isn't the only thing that comes out of here. So the main product is pyruvic acid. The byproducts are ATP and a molecule we call NADH. We're going to identify that here in a minute, or define that. So you can see not very effective so far, at least as far as making ATP. Only 2 are produced so far. So pyruvic acid will move on through the next process, which we call the bridge step. And this is going to convert that 3-carbon molecule into a 2-carbon molecule, which means we're going to release one of those carbons in carbon dioxide. And so this actually is the first place in this process, just like in our bodies, when we use glucose for energy, we breathe out carbon dioxide. This is where it comes from, or at least some of it. This is where some of it's being made. What that acetyl-CoA product is doing is feeding what we call a Krebs cycle, also known sometimes as the citric acid cycle. Now, the citric acid cycle produces NADH and FADH2. These are what we call energy carriers, and we'll talk about those in just a moment. But they're the thing that feeds, they are the substrate for the electron transport chain, which is where we're going to make the most ATP. Now, Krebs cycle also puts off a couple of byproducts. Some more of that carbon dioxide comes off, and some more ATP. But you can see so far we've got a net balance of 4 ATPs produced from that glucose. That's not very much when you're considering the millions of ATP molecules that a living thing uses over the course of 24 hours. So electron transport chain really ramps that up, right? So that one glucose molecule, if you were looking at the net response here, you have, or production of ATP, you have 38 ATPs coming from that one glucose molecule. Right? And of course, water is a byproduct of the electron transport chain. And where the oxygen comes from is going to be oxygen. The oxygen in that water molecule, it's coming from the oxygen that we breathe in, or that organisms are exposed to in their culture conditions. Under certain circumstances, the cell would be more efficient to use fermentation. And this is typically going to happen in the absence of oxygen. And so it looks just like respiration to start. It starts with the same substrate, starts with the same pathway, which is glycolysis, and produces pyruvic acid. Now, when fermentation enzymes are kick in, we're going to make those byproducts, the products acid and alcohol. We do have some byproducts as well, which are NADH+, which is actually going to be the substrate for that NADH that we used over in respiration. We're only going to make 2 ATPs through the fermentation process, because remember, a lot of that energy is actually lost in the acids and the alcohols. Carbon dioxide is also produced here, which is actually what makes alcoholic beverages bubbly, right? Bubbles in champagne or beer, that's where the carbon dioxide is coming from. Now, if you're talking about other carbonated beverages that don't have fermentation involved, those are artificially carbonated by industry. But this is basically the overall pathway. Now, keep in mind, glucose is not the only thing that living things can use to make ATP. So we're going to see where fats and proteins sort of slip into this pathway. Glucose is the preferred energy molecule. And so rather than making entire system of enzymes and everything that's needed to break down fats or proteins, we're really just going to sort of tweak those molecules to make them fit into this pathway. So keep that in mind as we move forward. Now, most of those reactions that are involved in what we just looked at require both oxidation and reduction. And so very oftentimes you're going to hear about redox reactions. So if you're the substrate that lost electrons, you were oxidized. If you gained electrons, you were reduced. And so the enzymes typically involved here are just referred to as oxidoreductases. And so the partnered molecules are called the redox pair. So this terminology comes out a lot. It's basically a lot of the steps in what we just kind of glazed over in the last slide involve this kind of reaction where you're just moving electrons from one molecule to the next. And so they refer to the oxidizing partner, the one that's donating the electron, we would call that the electron donor, and the one taking that electron would be the electron acceptor. And so this is where we're going to define those molecules that we called energy carriers. NAD, which is nicotinamide, adenine dinucleotide. Don't worry about that, just call it NAD. If you add a hydrogen to this molecule, it would be NADH. And the energy is carried in that hydrogen. Okay, the other molecule was FADH. That we saw in the coming out of citric acid cycle. And so FAD is actually a better energy carrier because it can carry 2 hydrogens at a time. So where those hydrogens are going, that's really the reason why we're sending these 2 energy carriers into that final step, the electron transport chain, which we'll look at in a minute here. So they call it the electron transport chain because you basically have a phospholipid membrane that has all these proteins in a sequence or a chain, and they're embedded in that membrane so that they're sitting exactly next to the previous substrate or previous enzyme. Essentially, they all function sort of as enzymes because they're in that process of multiple steps of metabolism to get to the result. So you can see here our energy carriers, NADH and FADH2, they're going to donate that hydrogen ion, and the enzymes that are parked in this membrane are basically going to put that hydrogen across, and we're going to build a hydrogen gradient. And so recall that hydrogen, when it's lost an electron, is all it's left is a proton. And so sometimes they'll call this a proton gradient. Once that hydrogen gradient is built, it's going to come back down through an enzyme that's embedded at the end of this chain that's called ATP synthase. Sometimes I've seen this as ATPase. It's just a little abbreviated version of the name of that. So obviously with a name like that, it's the molecule that is responsible for recharging ATP, right? So if somewhere in your cell ATP has been used for energy, basically what's happened is they've stripped off one of those phosphate groups. So this enzyme is responsible for putting that phosphate back on and producing ATP. It's really just a rechargeable battery if you think about ATP. And so how did ATP synthase have the energy to do that? You can track back here to that first enzyme. Right? It's actually when it takes the electron off of that NADH molecule to then leave that positive ion, hydrogen ion, on the other side of the membrane, it actually steals that electron and passes it to the next enzyme. That gives the next enzyme the energy it needs to run its reaction, and then it's passed along from enzyme to enzyme, and each one of those is capable of moving hydrogen ions across the membrane, which is, again, they're all contributing to that gradient. So you can see here at the end, oxygen comes into play here at the end. Look where that electron is actually finishing up or ending up. So very oftentimes the electron transport chain is referred to, it's sort of like a fire brigade, right? If you have a series of people that are passing buckets of water down to extinguish a fire. If you didn't have a molecule at the end that would take up that water, or that, in this case, the electron, you would never have a flow of this electron. It would sort of get locked up at that last stage. And so the thing that accepts that electron is oxygen. So you can see how we strip off some of those hydrogens that are coming off the ATP synthase. We mix that together with the oxygen, and that electron is what's driving that reaction. And so we make water. So there's the water being produced and the ATP being produced by what we call phosphorylation, right? So again, when you just add the phosphate back to that molecule, it's been phosphorylated. So where is this happening? Well, it depends on what kind of organism you are. In our cells, of course, we know the mitochondria is the powerhouse of the cell called the mitochondria. We have lots of membranes inside that mitochondria for this reason. Remember, if we're going to use a membrane to park all these enzymes right next to each other so they're very efficient, you need lots of membrane, which is why that inner membrane is folded and folded and folded. This is why the mitochondria is so good at making ATP. But if you're a bacteria, the only membrane you have is your cell membrane or cytoplasmic membrane. So that's actually where it's happening. Their ATP is happening right there on the surface of the cell. So at the end of that, we said that the oxygen molecule was going to take up that electron, and we call that the final electron acceptor. If the organism uses pure oxygen as that last step, which is really what the diagram we were just looking at, you were looking at an aerobic organism because that final electron acceptor was pure oxygen. Anaerobic environments or organisms can use other things. Yes, you can see there's oxygen involved here, but it's not pure oxygen. So again, sometimes if you're an anaerobe, it's because you don't have enzymes to break down toxic oxygen, or you're an anaerobe because you don't use pure oxygen. You might use something like nitrate instead of oxygen, pure oxygen. So again, we mentioned glucose is not the only molecule that your cells can use for energy. If we want to break down other molecules, we've got to just do a little bit of change to that molecule so it looks like something that fits into the glucose pathway. And so lipids, of course, lipids carry a lot of energy because of the way that they slip into the glucose pathway. So most triglycerides are the form of fats that we can digest and use for energy. So you can see here when we start, we have a series of fatty acid chains, 3 fatty acid chains attached to a glycerol molecule. The first step we would have to do is use an enzyme called lipase. And it's basically just going to strip that glycerol molecule off and leave us with lots of fatty acid chains. So part of that glycerol molecule, if you recall, we talked about 6 carbons to 3 carbons to 2 carbons. Look what we're left with when we strip off glycerol. It's a 3-carbon molecule, so it actually fits right into that one of those steps in glycolysis. So it can feed the aerobic respiration cycle once just by that glycerol molecule. But we've got all these carbon chains now that we can also use for energy. And so the next step would be, let's chop up these fatty acid chains. And to do that, we have to do a process called beta oxidation. And so you can see here the set of enzymes that would be used basically just chops that up into pieces of 2. And so if you look down here at the bottom, some of those molecules along the way are going to be produced that actually are the same substrate we need directly for the electron transport chain. But the rest of them are going to be chopped up 2 by 2, and they're going to feed that Krebs cycle because that's actually the basis of acetyl-CoA. If you recall, that's the substrate for Krebs cycle. So now go back here to the original molecule, count up how many 2 by 2 pieces of carbon you can chop up off of that fat molecule, and you can see why fats contribute so much more energy to the body than glucose, right? We're talking about each time you plug one of those molecules into the glucose pathway, you're going to get that 34 ATP at the other end of the process. So fats carry a lot of good energy. Cells can also use protein for energy. If you recall, proteins are basically a sequence of amino acids. And so in order to break them up into those amino acids, we need proteases. Proteases would basically break the peptide bond between each of those neighboring amino acids. Then, of course, because amine groups are still on those amino acids, the NH2 group is the amine group. That is not something that mimics those shapes of molecules that went into the glucose pathway. So we've got to strip that off if we want to use this molecule in the glucose pathway. So deamination would be the last step there. And look what the molecule looks like once you deaminate it, right? It's a 2-carbon molecule, which means it fits those enzymes that feed Krebs cycle. It looks a lot like acetyl-CoA. And so for every amino acid in that molecule, you'll get a round of ATP production as if it was one entire as if it was just one glucose molecule. So you can see why proteins and fats can contribute so much energy. For example, let me give you something for an example here. Albumin, which is the main protein in egg white. Why eggs are a really good source of energy or protein is because each albumin molecule is made up of about 585 amino acids. So you get to run that glycolysis pathway, the glucose pathway 585 times. And so, multiply that by 34 ATPs each round, and it's clearly providing lots more energy than a single glucose molecule. So typically metabolism requires all enzymes, substrates, all those things, but cells don't have everything necessary for every possible kind of metabolic reaction present all the time. They make and degrade these things as needed. So transport proteins, right? If you don't have the right substrate floating around, why have that transport protein taking up space on the membrane when that protein could be used elsewhere? Enzymes, same thing. It's mostly protein. Maybe some of those inorganic cofactors could be better used elsewhere. So they're only going to make enzymes that match substrates that they're actually exposed to. Metabolites, think back to that feedback inhibition. Everything requires more than one step, more than one enzyme. So why go 3/4 of the way through the process when you're not going to be able to finish it? So they're not going to bother with making metabolites or intermediate products unless they need to. So always the cells are going to use the most efficient molecules first, which are always the glucose molecules or the carbohydrates. Once they run out of that, then they'll bring up those other catabolic pathways as necessary.